First, I'd like to thank PacBio and the organizers for inviting us to speak at this event. It's been such a fun time getting to hear all the talks and hear all the complex and really fascinating genome assemblies. So today I'm going to tell you about how EpiCypher is helping to remove barriers to long-read epigenomics by focusing on a tech that you might have that's been mentioned in a few talks called Fiber-seq. So I'm showing you two pictures on the left side of the slide. One you'll recognize. This is our Revio that was installed recently. The picture on the far left is actually Hurricane Helene. And what these two things have in common is being a company based out of North Carolina, both of them arrived at the same time. So through heroic efforts by PacBio's team and our people on site, we got it installed. And with the help of Nancy and Manasi and Rhett, they trained us up on this system. And we're now using the PacBio SPRQ chemistry that we're really excited about. And so I just wanted to give you a summary of our first 9 SMRT cells. So our fragment size is about 10 kilobases. And so if you remember from David's presentation, these specs fit in line with what you expect from the really nice SPRQ chemistry results. OK, so a little bit about EpiCypher. We're an epigenetics biotech company. We have academic founders. And our philosophy is for scientists by scientists. And we really reach out to the scientific community through collaborations and with new emerging tech to enable groups to accelerate their research and elevate the impact. And so we have over 47 peer-reviewed publications. And so we really like to actively engage with the scientific community. And you'll see sort of that come to fruition by the end of the talk. OK, so a brief primer on epigenetics. You all know the classic Waddington model of epigenetics, the epigenetics landscape. So in humans, there's over 200 different cell types, all with the same genomic sequence. However, their transcriptional signatures are different, and this is driven by epigenetic features, okay, that drive development, identity, and when it becomes dysregulated, disease. 2 of the most commonly studied epigenetic features are DNA methylation and chromatin accessibility. And what I'm showing you here is enrichment that you would see for DNA methylation and its association with chromatin at the top, open nucleosomes or closed nucleosomes. And that's also associated with transcriptional activity versus repression on the right. And both of these epigenetic technologies are very popular. And so you can see these charts, they're still growing. The exponential growth is pretty astounding. And if you look on the right, chromatin accessibility actually had nearly 10,000 publications just last year. And this is primarily with short-read sequencing. OK, so what took the epigenetics field so long to embrace LRS? Well, there's really 3 things that come to mind. Number 1 is the input, because people in epigenetics often deal with DNA in nanogram amounts. So being asked to provide microgram quantities of DNA is kind of daunting. SPRQ chemistry really caught our attention in this regard. Secondly, cost. You can see over the last 10 or 15 years, there's a striking trend on cost per gigabase of sequence, to the point now where there's cost parity between the short-read sequencers and PacBio's Revio. So we actually have 2 NextSeq 2000s at EpiCypher, and we have a Revio at EpiCypher. So really, the Nova X is a bit of a mirage, because there's actually not that many of them placed, and we have access to the NextSeq. And you look at the price at the bottom, if we want to do 30x coverage, why would we pay twice as much now? So this is really exciting. The third thing is applications. And so I want to give a brief background on methyladenine transferases. So short-read sequencing technologies, they use enzymes that fragment DNA, because you want to obviously get the DNA to smaller sizes, which are required for short-read sequencing. And these are destructive. So thinking about moving into long-read sequencing with epigenomics, there's 2 key challenges. One is not fragmenting the material. And then secondly, if you're going to modify bases, not conflicting with existing modifications such as 5-methylcytosine. And so, as I mentioned, 5-methylcytosine is not ideal because it's common and it's commonly modified in mammals and plants. However, 6-methyladenine presented a really interesting opportunity. It's not in mammals and plants on DNA. Bacteria produce these methyltransferases. And a publication by Andrew Stragakis in 2020 showed that high of 5 is a really interesting adenine methyltransferase that outperforms others in its class. And so a really beautiful review by Anna Conesa, who's here, she gave a really nice workshop yesterday, summarizes what is being done in this space just over the last 5 years. So you can see that there's a collection of chromatin accessibility assays, and the one that performs the best is Fiber-seq. And putting it all together, you can see that when you do long-read epigenomics, of course, you always get 5MC for free, but then mapping protein-chromatin, protein-DNA interactions is there. But what I'm going to focus on today is chromatin accessibility and what you can really leverage with mapping open chromatin. Okay, so Fiber-seq was invented by Andrew Strigakis, and the assay works fairly straightforward. It's as simple as you can imagine. You isolate nuclei, you add the enzyme, wait 10 minutes, and you're done. So why would you not do it, right? You've got to isolate genomic DNA anyways, so it's really that easy. So the two direct measurements you get, of course, are DNA methylation, but also open chromatin. What is really exciting is between the open chromatin segments are what we call footprints or shadows, and so I'm going to talk about what these look like and insights we can leverage from there. And then, of course, you get all the genetics. I'm not going to focus on this because you all know all the genetic insights you get from long-read sequencing. And so with Fiber-seq, there's some challenges. The key enzyme Hi5 is not commercially available. There's no commercial support for protocols, so going from publication to a protocol, as you know, can be quite challenging. And then long-read sequencing can be relatively inaccessible. So we want to solve those problems. So what is EpiCypher doing in this space? Well, we actually have an early access program ongoing now. We've already shipped out this enzyme. We are producing the enzyme at commercial scale. We're characterizing the enzyme so that there's consistent lot-to-lot performance. We're optimizing the protocols to address questions such as, does it work in bacteria? Does it work in this organism? Can you use frozen material? So we're testing all of this at EpiCypher with our R&D teams. And we have plans to commercial release later this summer and also provide services and kits later. Okay, so we purified Hi5 at commercial scale, and we tested for disulfide bridges, and it's free of it. The purity looks great. And these are enzymes that modify the host genome's DNA are fairly challenging to purify, and so this is a bit tricky. The second thing we do on the right is rigorous enzyme activity validation. So what I'm showing you are two independent assays. The one on the left is a simple gel-based assay, where when we add Hi5, SAM, and DPN1, you get cleavage of the DNA fragment. So the DNA fragment is a defined PCR-amplified segment that has a single DPN1 cleavage site. And only when Hi5 is present with SAM does it cleave the DNA into two bits. The QC on the right is a much more sensitive and quantitative assay. It's kind of like FRET. I don't have time to get into how it works, but it essentially computes the EC rel, which is similar to, think of it like a KD. And so we can track this quantitative metric over time and from batch to batch to ensure consistent performance. Okay, so what does the data look like? We were very delighted. I said it was easy. Our first experiment, this is what the data looked like. So if you're not used to looking at chromatin accessibility profiles or heat maps, this is a heat map colored red to blue. Red is the 6MA signal, and the heat map is aligned to the transcription start site, the TSS at the bottom. And active promoters have open DNA or naked DNA just upstream of the transcription start site. So this is completely what we expect. When you look at an individual location, you see DNA methylation at the bottom, and then when you look up at the top, the green tracks, each individual row is a single fiber, which is a single 10 kilobase molecule. And you see that in the pileup at the very top, you see most of the chromatin accessibility is at the promoter, but you see all this green signal elsewhere, and that's not noise. That's real information. That's linker accessibility labeling and footprinting, all embedded in there. And we generate a lot of other epigenomic data. And so at the top, what I'm showing you are ATAC-seq, SRS, and this is the field standard. I showed you, the publications. What you'll notice is there's one peak at the promoter. You get nothing, very little elsewhere. So it's really not a good footprinting assay. It tells you where open chromatin is, and that's about it. You have to sequence really deeply to get footprints. There's a CTCF binding location right at that open promoter as well. K4 trimethylation is a histone modification that marks active promoters, and so that's consistent with it being open. And then on the left, the orange track is H3K27 trimethylation, and that's covering the body of the gene on the left, and that's K27 trimethyl is known to overlap repressed regions of the genome. So we can put all this together. So next, that was our first experiment. So next, we wanted to optimize the Fiber-seq protocol. And just in case you're curious, I have the list of published organisms Fiber-seq has been performed on: human, fly, plant, and yeast. All the data I'm going to show you today is with human K562 cells, which is a chronic myeloid leukemia line. So we did to optimize the assay, we did a simple enzyme titration, and you can see that we're getting increasing 6-methyladenine labeling as visualized by that heat map. When you look at individual genes, you can see from top to bottom, low concentration to high concentration. The intensity of the green increases as you add more enzyme. So this is a tunable assay. And so the question is, how do you know whether you're over or under labeling? This is also a problem in ATAC-seq, the conventional open chromatin assay. So what I'm showing you here is tracking the total 6-methyladenine on the y-axis with the reaction conditions on the x-axis, so the reaction time. We perform the reaction at 2 temperatures, 23 and 37 degrees, and you see the labeling going up. So what is the right amount of labeling for human cells? Well, it turns out that if you track the nucleosome sizes, so those footprints between the linker DNA, you can see that the more you label, the smaller the apparent nucleosome size becomes. And because we know that, and if you put these two metrics together, nucleosome length on the y-axis and 6-MA labeling on the x-axis, and we know from crystal structures, biochemistry, and MNA-seq nucleosome mapping papers, the nucleosome size is 147 base pairs. And so if you draw a horizontal line, that gets you the optimal labeling. So we think this is a rubric that can be applied to different organisms to determine what the optimal labeling is. Okay, so let's talk about what else can Fiber-seq do. So that's just 5MC, 6MA labeling. Okay, it's great, we got open chromatin. But what about those footprints? Well, in the genome, there's also, in the human genome, there's 15 million nucleosomes in the haploid human genome. There's over 2,000 transcription factors, sequence-specific transcription factors, several hundred of which are active in a given cell type at a given time. Well, now I'm sort of wondering what a gene is, but the 20,000 is the human protein coding gene number that's typically cited. And RNA polymerase is transcribing many of those protein coding genes, right? And then lastly, enhancers are elements that help to enhance transcription. And so there's 3D contacts that are made. So how can Fiber-seq inform on these different epigenomic features? So I wanted to give you a high-level conceptual framework for how this is done. So we have patches of 6-methyladenine. Imagine sort of a segment of the genome. And you have these patches of varying length. They typically come in 2 varieties you see at the bottom. Open chromatin is usually around 125 base pairs, and then linker DNA between nucleosomes and other features is usually pretty short, about 50 base pairs. But then what's between it is really exciting because it's footprinting nucleosomes, it's footprinting transcription factors, and polymerase. And so we can bring in annotation, of course, where the gene introns and exons and start sites are. Because sequence-specific transcription factors have known characterized motifs, we can bring that information in and ask, well, what are we seeing here? And then there's other databases as well, short read tracks and other things. Okay, so what do the inferred nucleosome positions look like? So this is a heat map of nucleosome positions, and you see that it's almost like the mirror, the inverse of the accessibility, where instead of having the signal at the promoter, you have this void of signal at the promoter because that's naked DNA. And then you see this really nice, to the right of the transcription start site is the +1 nucleosome, and then you see this periodicity of the nucleosomes emanating from it. And we have hundreds and hundreds of short read data sets, and I talked about K4 trimethylation. It's linked to active promoters. K27, it's linked to inactive promoters. And you see when we just align that data, it fits perfectly. So it's predicting the activity of the promoters. And when you look at an individual location, I'm zoomed in a little bit here now. So this window is 2 kilobases. And what you'll see is the DNA methylation patterns at the bottom. And at the promoter of NUP214, you see really strong, broad, open chromatin. But if you look to the left and to the right, you see the beautiful periodicity of the linker DNA. And when that algorithm FIRE calls the intervals of regulatory elements, this is what you see. The nucleosome positions nicely placed between the accessible regions of the DNA. Okay, so what else can Fiber-seq do? So CTCF is a really well-studied and well-characterized transcription factor. It's also called an insulator because it helps to guide 3D contacts of the genome. And so on the left, what you're seeing is the 6-methyladenine signal colored in red, and it's aligned to the CTCF motif that's at the top. It's aligned to the midpoint of that motif. And if you look closely, you see this blue stripe all the way down, 80,000 locations. So without doing any more sequencing, we're getting the position of 80,000 CTCF binding locations. Okay? So this is what it looks like. This is one location. Of course, you have your individual fibers at the bottom. Green is 6-methyladenine. You see 2 big accessibility patches right in the middle. And just above it, there's a track of the CTCF motif that's nestled right in that accessible region. And then for validation, I have 2 tracks, CTCF, cut and run. So when you do a standard chip-like or cut and run assay, you see that there is real functional CTCF binding there. And of course, it's open as well, as indicated by the ATAC-seq track. Okay. If you look at a more complex location, there's 3 CTCF motifs at this location. And what you'll notice is you can see the heterogeneity from left to right. The leftmost motif is about 50% bound. The middle motif is the main position for CTCF occupancy. And then on the right, it's kind of in between. And so you can look at molecule-to-molecule heterogeneity from a vertical slice, but you can also look at a horizontal slice. So within a single 10 kilobase fragment, that red box at the bottom, you can look at an individual fragment that's coming from an individual cell and asking what is the co-occupancy of CTCF and other features. You can really put it all together in a multi-omic fashion. Okay. Something else that hasn't been talked about is inferring Pol II locations. And the way this works essentially is, as you can imagine, up top there's a cartoon showing polymerase that's transcribing genes. And Hi5 will then have access to label the adenines that are unprotected. And so what you're seeing on the bottom left are those individual single molecule fibers lined up. Green are nucleosome-sized positions, and then you see that white void. That's where polymerase sits. And then on the right is a shadow. It looks more like a footprint. So you see a white void at the top part of that plot. That's actually polymerase that's already initiated transcription that's just to the right of the transcription start site. And then the white footprint at the bottom that's a little bit lighter, that's pre-initiated polymerase. So you can see both phases of polymerase coming to genes and then initiating. It's really neat. You saw some of these maps in the last few days, so I'm not going to go into too much detail, but enhancers make long-range contact with promoters. The point I want to make here is not only can Fiber-seq predict these contacts, but if you look at the metrics I have on the right, this is a high-C assay. So high-C is just what they call the proximity ligation-based approach to map contacts. 600 million reads are needed to generate high-C data. And it's really important for the resolution of the contacts. That's 120 gigabases. So why would you do high-C if you can do Fiber-seq for the same depth with long read and get nucleosome positions, chromatin accessibility, DNA methylation? It's really a fantastic multi-omic approach. Okay, so just bringing it back, EpiCypher is removing barriers to PacBio long-read sequencing and epigenomics. We're supporting enzymes, assays, engaging with the community, and so stay tuned. And if you're interested, we've begun our early access program. I put my email up here. If you want some enzyme gratis, all we ask is feedback on how it's performing in your organism of interest. You don't have to tell us anything you're not comfortable, But yeah, with that, thank you for your attention, and I'll take any questions.